Hello, and welcome to part 3 of our lecture series on the urinary system. And in part 3, we're going to continue on with our discussion of what's occurring within the nephron. Now, essentially, we had taken a look at what's occurring to our raw filtrate as it's passing through the proximal convoluted tubules, and we saw that we're essentially going to be looking at cells that are going to be actively reclaiming things like sodium, like water, like glucose, amino acids, things like that, and getting rid of materials that the body is going to have as waste materials, like ammonia, like hydrogen ions as an acid. The next thing we're going to look at is that proximal tubule is going to go into the loop of Henle. And the loop of Henle is going to be a U-shaped epithelial tube. And it's going to be composed of, again, relatively straight tubules. And so it's going to run relatively straight down into the medulla, twist back around a hairpin loop, and come back up, back towards the cortex. And both the cortical nephrons and the juxtamedullary nephrons are going to have the same overall appearance. But from a physiological perspective, we're going to focus in on those juxtamedullary nephrons. And the reason for that is that they're going to be very important for establishing the very high osmotic concentration within the medulla. And it's going to be important for producing concentrated urine later on. And so we're going to be talking about the juxtamedullary nephrons, even though they're only a small percentage of the nephrons within the kidneys. And so what we're going to be looking at is the loop of Henle as a U-shaped epithelial tube. Now, if we take a look at this, within the loop of Henle, we take down within the medulla, what we're going to see is that we got these straight tubules. They're going to be packed in very close to one another. And because they're straight, they're going to be running all in the same orientation. So it's not like going to be in the cortex where we have the convoluted tubules, everything's twisted around. Sometimes you see a longitudinal, sometimes a cross-section, sometimes we can't tell what orientation it's in. All of these are going to be in the same orientation. So they're all going to be cross-section or all going to be longitudinal. Two portions of the loop of Henle, kind of anatomically, that we can take a look at. Thin portions where we're essentially going to be looking at cells that are simple squamous. The nuclei are bulging into the lumen, and they're going to have a lot more cytoplasm than capillary cells. And it tells us that we're looking at an extension of the uriniferous tubule. Or the thick portion of the loop of Henle, where we're going to be looking at generally cuboidal cells or maybe low columnar cells that, again, are going to be pretty distinct as being a portion of these uriniferous tubules. Both the thin and the thick portions of the loop of Henle are going to have about the same diameter of the lumen, but the only difference is essentially the thickness of the wall of these structures. Now, before we go on, I want to review osmolarity because we're going to be talking about the functions associated with the concentration of materials. And so with osmolarity, we're going to be talking about isotonic, hypertonic, and hypotonic. And again, this is going to be a relationship to one another. And so isotonic is in essence going to be the same concentration or same tonicity as the normal tissues or the normal blood. And so on this diagram, isotonic is basically what's going on within the bloodstream. Hypertonic is basically going to be hyper, or essentially higher concentration or more concentrated. And so we're going to have more materials packed in it, packed in the same volume, than we would do within an isotonic region. Isotonic, again, the same as what we can find in normal tissues and normal blood throughout the body. Hypotonic, hypo is going to be more dilute, and so we got a lower concentration. And so when we take a look at this within the kidneys, what we're going to see is that within the cortex, we're going to be dealing with isotonic. As we go into the medulla, basically with that corticomedullary junction, the junction between the cortex and the medulla, we're going to start out as isotonic, and then it's going to become more hypertonic, more concentrated as we get closer to the tips of the medullary papillae, the tips of the medullary pyramids. So the deeper within the medulla we go, the higher concentration, the more hypertonic we're going to be looking at. Now, if we take a look at this, we're going to take a look at the loop of Henle, and this is the diagram kind of twisted over on its side. We've got the descending limb of the loop of Henle, essentially going on the top, going from left to right. We got the hairpin turn, and the ascending limb going along the bottom from the right to left. And it's essentially going to be what's referred to as a countercurrent multiplier. Countercurrent means that we're going to have materials that are flowing in one direction and then they're going to turn around and flow back. And so again, that hairpin loop that we're looking at here with the descending, the loop, and then the ascending portions of it. And as we're going to be going through, we can take a look at the concentrations that are present. And again, close to the left-hand side of the slide, we've got 300, which is roughly isotonic. And then as we go towards the right-hand side, we're going to be going deeper into the medulla, closer to the tips of those pyramids that we talked about. And we're going to be looking at a more hypertonic, a higher concentration of materials, both within the loops of Henle, as well as in the connective tissue, the spaces around them. So if we take a look at this, what's going to happen within the loops of Henle is we're going to establish that osmotic gradient. And so we're going to look at what's occurring within the descending portion of the loop of Henle and then the ascending portion. And so within the descending portion of the loop of Henle, we're going to be looking at a structure that has basically a passive role in establishing the hypertonic interstitium. Within the descending portion, we're going to be looking at a structure that's going to be permeable to water and impermeable to salt. Okay, so we need to think about what this means. Impermeable to salt basically means that ions and materials within that raw filtrate, within the fluids that are passing through the descending limb of the loop of Henle, the materials, the solutes, are going to stay within it. It's going to be permeable to water. The linings are going to be permeable to water, meaning that water is going to flow out. And again, keep in mind that what water is going to do is through the process of osmosis, it's going to try to balance out the concentration. So if we take what would be isotonic up at the top of our medulla, and we're going deeper into the medulla, going down into a region that is a higher concentration, water is going to flow from the inside of the loop of Henle out into the interstitium, out into the tissue space to try to dilute out the materials that are there, to decrease that hypertonic, that higher concentration by adding volume, by adding water to dilute it out. But in doing so, what's going to happen is that the water is flowing out. What's going to happen with inside, the materials inside the loop of Henle, is you've got the same amount of solute, the same amount of salts, but a lower volume. And so we're essentially going to be concentrating the materials inside of the descending limb of the loop of Henle because we're essentially almost like evaporating the water. The water is leaving the inside because it's going out into the interstitial space, into the tissue space. So what's going to be left behind is a smaller volume with a higher concentration. And so we're going to have towards the bottom of our pyramid, before we make that hairpin turn, we're going to have a higher concentration of materials in the descending limb of the loop of Henle. Not because we pumped things, not because we did anything, but because water simply flowed out through the process of osmosis and left behind our salts, left behind our solutes. That we've got a smaller volume, same amount of materials, but because we've got the same materials in a smaller volume, it's going to become more concentrated. Now, if we take a look at what's occurring on the ascending limb of the loop of Henle, we're basically going to be looking at something that's opposite to the descending limb. The descending limb, we said, was permeable to water, impermeable to salt. The ascending limb is going to be the opposite of that. The ascending, the portion going from the deep medulla, past that hairpin loop, back up towards the top, is going to be permeable to salts and impermeable to water, meaning that the water that is there can't flow through the process of osmosis. It can't flow out into the interstitium, and water can't flow from the interstitium into that loop of Henle, into the ascending limb of the loop of Henle. But what's going to happen is we're going to low volume at the tip of the loop of Henle. As the materials are passing through the ascending, as they're being pushed up back towards the cortex, we're going to be not changing the volume, but we're going to be changing the salts. So we're going to be looking at the situation where we're going to be pumping chloride ions from the filtrate, from the inside of the loop of Henle, into that space, into the interstitial space between them. But we're essentially taking chloride ions out of the loop of Henle, out of that filtrate, and then pumping them outside of that. In doing that, sodium is going to follow passively. So we don't have to pump it along. Chloride's going to go, sodium's going to follow it. But what's going to happen is we're going to have the same volume, but we're going to start to take the solute. We're going to start to take the sodium and chloride out of it. And so we're going to reduce the concentration at that point because we're taking the materials out. We're taking the solutes out within that region. So what was very hypertonic, very concentrated within our loop of Henle at the tip, as we're coming up the ascending portion, we're essentially going to dilute it out again, but essentially not dilute it, but to decrease its concentration by pulling out the solutes, by pulling out the sodium and chloride, and bringing it back to either an isotonic or in some cases even a hypotonic, a lower than concentrated, lower than normal concentration of fluids within the ascending limb of the loop of Henle. Now, the ascending limb of the loop of Henle is going to allow the materials then to flow into the distal convoluted tubule. Distal because the filtrate, the materials within these uriniferous tubules have been processed and flown through the proximal tubules. They've gone through the loop of Henle, the descending, the ascending portion. They've gone a long ways, and now we're going to be back up into the cortex, into the distal, and now convoluted, they're kind of twisted around. We take a look at the epithelial lining. Again, low cuboidal epithelium, no brush border because we don't have microvilli along the apical surfaces here. And because of that, the lumens are going to appear to be much wider. We're going to see more nuclei within the area than we would with the proximal tubules, but we're still not going to see clear boundaries between the cells. A little bit more basophilic staining appearance to the distal convoluted tubules than the proximal convoluted tubules. Smooth surface, more nuclei, and that's the characteristics of the distal convoluted tubule. Now, if we take a look at these cells, aldosterone causes these cells to essentially remove more sodium from that raw filtrate and replace it with phosphate. And so we're essentially modifying these materials as we're going through. And that's going to finish up our look at the nephron. We've got an additional structure that's found within the structure, but it's not directly related to the processing of the urine. And that's going to be the juxtamedullary apparatus, which we're going to look at in part 4. If you have any questions, feel free to email me at hoffmanj@arcadia.edu, and hopefully come back for part 4 and hear about the juxtamedullary apparatus. I'm sorry, the juxtaglomerular apparatus.